Okay, we should be going live.
Awesome. So I think that we should start without further ado because we're already quite a few minutes late. Sandy, you want to start something, or you want me to introduce?
I was just wanting to say that okay, we should be going live.
Awesome. So I think we should start without further ado because we're.
So you're getting the feedback from the audio. It was just to say that we met. I don't know how many years ago now. 20 years ago. How long has it been?
Not that 20 years, I think, but quite a few years.
it was during the ICBP, right?
Yeah.
And at that time, there wasn't a lot of people doing mathematical or computational work as part of the program. And so it was like, you know, someone else here actually understands what we're trying to do. So it's been great to watch your trajectory. And even if you're kind of maybe coming from a more chemical biology sort of background, you're still using mathematics and modeling and data and sort of driving novel insights. And we really appreciate that. So we're looking forward to hearing what you've got to say, but I'll let David formally introduce you anyway. It's great having you here.
Yes, I'll try to be brief. Not that I have really reasons, just because your CV is very spectacular. But obviously, Professor Xing, who you can see on the first slide, is at the University of Pittsburgh, where he is a professor at the Department of Computational Synthesis Biology at the School of Medicine, and he also has a joint affiliation at the Department of Physics and Astronomy at the University of Pittsburgh. He got his bachelor's degree from Peking University in chemistry. Went to the United States to do to join grad school, where he got a master's in chemical physics in the University of Minnesota Twin Cities, and went on to do a PhD in theoretical chemistry at the University of Berkeley, where he stayed to do a postdoc. Then joined the research laboratories at the Lawrence Livermore National Laboratory. Became faculty member at Virginia Tech, where he was assistant and then associate professor, and then moved to Pittsburgh, at the University of Pittsburgh, where he is currently obviously a member of the faculty. He has a nice series of papers, many of them very recent as well, in places like eLife and Cell, which I'm sure we'll be hearing some about today. And he has very interesting research where he asks the questions of how cells maintain and control their phenotype, and he's using single cell data to do that. So, Professor Xing, without further ado, please take this over.
Okay, thank you, David, for the introduction. And thank you, Sandy, for inviting me to give the talk. It's really, I think, I'm really excited to share our recent work with the general audience. And a title suggests is. Our work is reconstructing cellular dynamics from single cell data. So as David just mentioned, my background is chemistry, chemical physics. So because of this, in chemistry work on how chemical reaction takes place. So I got very interested in this, the problem in biology is cell phenotypes. So what's shown here, we know in our body, there are many different cell types as shown here. You can tell they are very different. They function are very different. They have different morphology, expression, all things. Then we also know cells can change phenotypes. This happens in developmental processes. Importantly, shown here is called this epithelial to mesenchymal transition. You can tell the cells are very different. So under some stimuli, cells can change from one phenotype to another phenotype, or even go backward. So we call it EMT or MET, the mesenchymal to epithelial transition. It happens during the developmental process. It happens in some pathological process, like this cancer metastasis. So cancer cells will hijack this process, so can disseminate it and form secondary tumors. And also happens in fibrosis. Another example of cell phenotype change is cell reprogramming. So we all know that now people have shown you can reprogram a terminally differentiated cell like a fibroblast into either this pluripotent stem cells or can directly go to some other cell phenotypes. So those processes are very fascinating and also biologically very important. So we are really interested in how cells regulate their phenotypes and how to control the cell phenotypes. So you can tell, right? This is a very important biomedical implications like a regenerative medicine and also in cancer treatment or other things. So for biologists, there are many tools have been developed. So here just show some examples. I show here, people can use the genomic tools like CRISPR for the gene editing. There are imaging tools like shown here, either this bright field or some fluorescent labeling, fluorescent imaging. There are many other tools. So those are the tools being developed by biologists to study those processes. But on the other hand, I want to tell you this, we know that in many fields of science, engineering, we have frequently used those tools, language to mathematics to describe process. Like shown here, this story is pinball machine, right? So then we know that supposedly for this system, we know this system, lot of parameters, their interactions. We can write down a set of equations, like shown here is the core, position, and also its momentum. Then we can write down the mathematical equation. We can describe or predict how the system evolve over time. So we know this is important development in physics. So it happens several centuries ago. I would like to review the process, review the history, just remind you guys. So we know that's first of all, there's some researcher like Jacobi. He spent all his life trying to collect the data, let's say the planet motions. Though this is really big data in that age, right? In that time. Then his student Kepler, he really summarized, worked out a set of empirical rules, so can view this kind of data mining process. But at the end is where Isaac Newton comes. He developed those theories that can describe those planet motion. So with this theory, now we can go beyond this data mining. Instead, we can also, the set of rules, the principles we can use to describe many other things. Specifically, as shown here, is molecular dynamic simulation of this protein, how the protein moves. Okay, so this is how physics have been developed to use mathematics to develop mathematical theories. So with this, I want to ask this question. Okay, for cells, we know cells also can cellular processes. Can we also write down a set of equations to describe how cells evolve over time, cell states evolve over time? So I show here a very generic one. So we have Z. Z is some variable, this specifies cell state. Then it will evolve over certain time, certain functions, and also here we use generic, we use some noise terms to specify, describe all those degree freedom not resolved for and will have impact on the system. So now the question is definitely how to specify the Z, right? And also how we can find out this function form of A. Okay, so here you may think, first of all, okay, this is really now something in biology, but then you realize, we have utilized this kind of language in biology for many years. So this is in the field of systems biology, mathematical biology. For example, I show here in gene regulatory network. So let's say we have a system. So we want to study how the system evolves with time. So we know it's regulated by a set of genes. And so we want to know how systems evolve. So we know what is the cell state. So it's what is now and how it evolved, right? That's how we want to specify cell state dynamics. Let's say for simplicity, talk about two genes. And, after many studies, biological studies, now we collect the information. We say, okay, there are two genes that specify for this process, just schematically, let's say. So then we know there's a self-activation of this gene. So then we can write down, equation expression form to specify this self-activation as shown here. So you can see the function form like this. So X is the expression of X1, gene 1, and see this function goes up with X1, reflect fact that's a gene, this is a self-activation, and also eventually saturate, right? So then we say there's gene 2 also have self-activation. So you can also write down similar function form for that. Then there's a mutual inhibition. So we can also write down a mathematical expression to reflect these biological observations. So this function, it has this form. So it decreases with this argument. So reflect this inhibition. Then you will have a degradation term. So you see, the idea is we have biological observations. We use mathematical equations to kind of translate those biological observations into mathematical equations. Then we can do analysis. So because with these equations, so mathematically, we say we have X1, X2 that can specify cell state at the instant time. And so we can form, we see the state space. Then the XDT is equals vector field. So it means for any point, you have state to tell me the cell expression of X1, X2, I have arrow. The arrow specifies the direction and the amplitude. So the system want to move. So there, if we know the information, so then we can propagate equations. Then we can predict cell behaviors. And especially we can do analysis like here, see there's attractors. So means those cells, they tend to go those attractors. That's corresponding to stable phenotypes. So this is how we typically do in mathematical modeling with biological networks. So we need to collect the biological information and quantitative data. Then we try to write down equations, try to estimate the parameters. Then we do analysis. So this is a typical procedure. So, but we know a network, a biological system is never as simple as this one, right? So many genes, they interact with each other. So this simple motif that really embedded in a large network. So I want to ask question is, can we reconstruct the genome-wide vector field? So help us to discuss how the system evolves, how different programs interact with each other. So you may think, okay, they really mission impossible. Since because even for this simple motifs, already take a lot of efforts to estimate the parameters, to determine the function forms, and do and then analysis. So how can we go to genome-wide? So the turning point comes when postdoc, Dr. Xiaojie Qiu, he is a postdoc at Jonathan Weissman Lab. So he approached me, he showed me a paper. So that's how we started the collaboration. And the work I'll show is mainly done by the two very brilliant young researcher, Xiaojie, and my the student Yan Zhang, as co-advised by me and my colleague Yvette Bahar. So this is the paper, and you can see it's really a collaborative research among many researchers. So let's go back to this. First, go this the paper Xiaojie just showed me. So this is a paper published a few years ago. This is a very seminal research. We know when we do a single cell sequencing, we have to destroy cells, so we only get snapshots. But those people, they say, okay, actually you get some information, dynamic information from single cell RNA-seq data. So the idea is consider a gene. You have we know a gene got transcribed, got to go to unspliced mRNA with introns, and then splicing, remove the intron. So this can be used for translation. Then the splicing mRNA got degraded. So mathematically, you can view this as kind of a rate process. You can write down some equations and write down some with rate constants to characterize those processes. Then those people suggest actually, this unspliced mRNAs, you can actually read the information from the data, because this is have intron, this does not have intron. Then they suggest if there's a way we can kind of estimate this rate constant, specified gamma, the degradation rate, and you for simplicity assume the splicing is 1. So that means they talk about relative dynamics. So O relative the splicing rate, then you will be able to get this estimate, the SDT, so called instant velocity, right? They call this R velocity. So this is the paper, and sounds very simple, but surprisingly, it works pretty well. So we want to go further. So we realized one thing, so if we have to make this kind of mathematical consideration, we have U, the unsupplied and the supplied RNA, right? So we know this transcription is really a function of also many regulatory elements, right? The expression of elements. So it's not really constant expression of many elements, and so that reflects gene regulation. Okay, so if we assume there's U and S, they're kind of redundant. So S is already complete for a specified cell state. So we really make this Markovian dynamics, right? So all the dynamics really can be approximately determined by what's the current state. So in that case, then U we see as a function of S. So we have a function of equation of S. So that's the equation of motion for the system. So now the question is, how can we determine this function? We know this function, okay? So this becomes a machine learning problem. We have a lot of single cell data, but each data we can read out this X, the expression, and estimate the XDT. So we want to find out this vector field function. So this F is a nonlinear multidimensional vector function. So it's become a machine learning problem. Then we, but there are certain challenges. The first challenge is, well, this original R velocity, you give you some quite a decent estimation, but can we go beyond that, even more accurate estimation, help us to learn? Because there are some limitations with this kind of splicing-based input. It's ill-defined for splicing and splicing. Because splicing, think about splicing, it's really a sequence of processes. So the splicing connect can be complicated. So and also bias or specific transcript, because different genes have different different amount of introns, and for those organisms have no introns, then it won't be suitable. And also it's relative, since we everything relative to beta, the splicing. Can we go beyond that? So this is Xiao Jiekam idea. So he noticed that there's a method people have developed to try to kind of measure the mRNA turnover rate, turnover dynamics directly using what is a metabolic labeling method. So I won't go those in detail. The idea is you treat cell with some chemical that induce certain chemical modification, and then some mutation, and the mutation will be captured by sequencing. So therefore, because of this, so you can distinguish cells, the transcripts, old and new, right? Because before treatment, all the transcripts that be without this labeling. So from there, you can infer the mRNA turnover dynamics. So based on this method, then this is we have developed a kind of procedure, learning procedure, so we can for different types of data, single cell data. You may have labeling, you may not have labeling, and different strategies, different schemes for getting the data. So we develop a set of methods that can infer the mRNA turnover dynamics and got the estimation of R velocities. Here I show you this. With metabolic labeling, we can go beyond this relative one. We really got the true dynamic, the real dynamics. Here's one example. So this is we analyze the data for one is the mouse stem cell, embryonic stem cell. The other is the human cells, REP1 cells. So we look at a set of genes. They have the mouse genes and also their human orthologs. And consistently, you can see this is a degradation rate. This is for this splicing rate. Consistently, the mouse genes have faster dynamics than the human genes, which is not surprising. We know, right? Because mouse have a shorter lifespan, and we do expect them to have faster dynamics. And those kind of observations have been obtained in some previous studies. In those studies, they focus on specific genes, they measure them, they have this conclusion. What I want to show here is from single-cell RSeq data, we were able to get this genome-wide characterization of this observation. Okay, so now you have this discrete samples. We want to learn this function f. So this is become a, since we do not know the function form f. So it just means you really try to find out a bunch of possible choices. You try to find what's the best one that can give you. So we're really working on this vector function space. So, so mathematically, what we do is, so this is what you do is you really have a bunch of functions. So you really express your all the possible candidates as a linear combination of some basis functions, and then you try to find what's the best function. So it's mathematically for the Hilbert space. So let me just, instead of going to detail, I want to give you a kind of very intuitive discussions. So how we define the function? I think you already have done those data analysis like fitting. Suppose you have data, X and Y. So you want to fit, for example, a linear curve. So you do this, right? You have tried to find out what's the best fit to give you the coefficients, the constant coefficient and the linear coefficients. So you may go even further to nonlinear fitting. So you got actual terms. So then you try to get the best fit. So here, what we do is really go to more general. This is 1x squared. Those are kind of basis functions you can think about. They form a function space, the basis of function space. So in general, you have a basis function. Each function can represent a linear combination of this. Then you just try to find this coefficients. So this is the basic idea. The procedure is like this. We have data. This is the data from single cell genomic data. Then we try to find what's the best function f, right? So we expand it by this basis function gamma. And then we try to find the best fit that will minimize the difference between the measured velocity and we calculate velocity. Then you have this normalization, regularization factor to penalize those overfitting, right? So this is the general procedure. And at the end, you don't need to worry about all the details. The end is we will have analytical function form of vector field. It's in high dimensional space. Typically, for we found out in 20, 30 principal component space will be sufficient to describe a set of data. And this each PC mode is a linear combination of the genes, right? So really, we're talking about the Genomewide space. Then we can do further analysis. I'll show here. So I'll show you one example. This example is about human. It's a hematopoietic differentiation system. We know the stem cells differentiate into different blood cell types. And although it's very well-studied system, there's still a lot of open questions. So the data here show the data we obtained by our collaborator at this Broad Institute in Harvard. What they look at, they use is human CD34 positive hematopoietic stem and progenitor cells. At different time, they collect cells and do this sequencing with metabolic labeling. Here, show you, this is the, you see in this UMAP space. And this arrow represents the cell, the velocity, I mean, the direction the cell want to go next time. And you see with this labeling, clearly the velocity is much smoother than this with this original splice-based velocity. More importantly, you look at those cells. So we know the cells were differentiated from progenitor to this differentiated cells, but this for the splicing velocity got the wrong result, wrong direction. And we have tried different packages, but always get the same thing. So it's not because of the analysis, really some inherently wrong with the data. Well, for this the new type with metabolic labeling, we got the right direction. Okay, so here show you, then we have this, got the vector field. So this is a vector field shown in this two-dimensional space. So you can see you can have these attractors. Those attractors correspond to stable phenotypes. This the repulsor, unstable points. This is a stem cell because we know this spontaneous differentiation. So this is unstable. Cells differentiate different phenotypes. You can follow, you can get the lineage trees. Since this is a vector field, so just like you have a force field when you do molecular dynamic simulations, you can really run simulations. So here show you you can run simulation for individual cells, how they differentiate. So here shows the movie. Okay, so now you have the vector field. How can we gain some mechanistic insight? Here I will show you, use this toy model to show how we can get it. So this is the two genes mutually inhibit each other with self-activation. I just talked about at the beginning. So this is called the toggle switch. So here I show you, for this toy model, we do have this underlying vector field. So we know if you have this vector field, if you do this partial derivative, they call it Jacobian. So this one, like shown, for example, that's this one. So experimentally, you can view this kind of experiment. It's like this. If you do the experiment, you upregulate slightly, upregulate the X1, the gene 1 expression slightly. You ask how it will affect the transcription rate of this one, so gene 1, right? So here show you is a positive, and it's increased. So this is this is activation. So this is another one. Is if you upregulate gene 1, then the transfer rate of gene 2 got downregulated. So this is negative, irregular it's a repression. Okay, so now let's look at real data. For this hematopoietic stem cell differentiation process, what I shown here, two well-known master regulators, GATA1 and SPI1 or PO1. So we know these two master regulators for this differentiation process. And decades of studies has established that the two genes, they mutually inhibit each other, and there's also self-activation. So it's a very well characterized toggle switch. And I show here is from the data, you look at the Jacobian and see this is blue is negative. So it's inhibition, self-activation. Okay, more than that, right? We can also get quantitative information. So this is the same data. We just plotted differently. This is the SPI1 count, and this is the heat map of this partial derivative. So from there, we can get a fit. Then we can just do an integration to get this F function. So this is just the dose response curve, right? What we just fit it with this Hill function we typically use in system biology. Well, then we found out for this SPI1 inhibition GATA1, we got this nice sigmoidal shape as we expected with Hill coefficient about 2. This is what we expect, right? What surprised us is this GATA1 inhibition on SPI1. So we got this Hill coefficient 1. So this is, at first we were kind of disappointed because for this system, there are tons of mathematical model studies on the system. We always assume the Hill coefficient has to be more than 2 or more for certain mathematical properties, right? But now we got this n equals 1. So is there something wrong in our analysis? Then we found this paper. So this paper, what these researchers, they really want to quantify their regulation. So what they do is they construct a vector. So you see this vector have the luciferase reporter, have this P1 promoter. So when P1 bind, well, generate the luciferase signal. And they transfect this vector, a second vector expressing P1, a third vector expressing GAT1. Then they transfect with different amount of GAT1. They look at how this P1 transactivation activity will be affected by GAT1. So we see this inhibition, and we see also there's, as they reported, there are no apparent thresholds, because we know if you have a sigmoid shaped one, you have this threshold. So this is really, got really excited because you see this, even if you're not experimentalist, it takes a lot of effort to do this experiment. But now I show you with a single set of well high quality single cell sequencing data, we can get quantity information of this regulation. And also not just these two genes, we got genome-wide, those gene regulation relations. So that's what the exciting part. You may also ask, why you have this asymmetric relation? Can we get some mechanistic insight? Here I give you some very hand-waving argument, since we haven't done experiment. So
can I ask a quick question?
Yes.
Sorry, could you go back a slide, couple of slides? One more. Yeah, this one. So this is very cool. I want to make sure I understand it. Are you saying, so I see this as a relationship between those two genes, but you're saying it's genome-wide. Is it genome-wide because it's those two genes in the background of all the other genes? Is that right?
Okay, that's a good point. So first of all, yeah, when I got this, I do have average over genome-wide background to get this. I can definitely get more, more specific context-dependent things. When I say genome-wide, I mean, I can, from this same set of data, not just the GAT1 P1, I can get other genes that I have read in this genome, other transcription factor, other genes, their gene regulation relation. So that's right. What I mean.
Yeah, okay. But obviously you're looking at two genes in the context of the fact there's many others that are also regulating those two genes, right? So
yes,
which is fine, but I'm just saying there's a whole picture we don't see here.
Yeah, okay.
Right.
I want to tell, when this is, when we have the gene vector field, we have all those informations, right? So I want just to dissect it to get something information or more familiar to the system biologist. So this is so when we say this F function really averaged over the genome background, I can have more than that, right? Because of those in the vector field, I can have, okay, what the third gene, how that will affect me. You see, this is a very, that's definitely the background. So we do have ongoing work. Because the reason I do this is when we construct a vector field, we use this Hilbert space, right? So we have a basis function. That's for mathematical convenience. But then you lose this interpretability, right? It's not clear to know. So we do try work on a different way. We use those basis function we're familiar with in system biology, like Hill functions. So we can, that one will have better, more apparent transparency on the gene regulation relations.
Yeah. Okay, cool. I think it's great. I'm intrigued about the relationships it's uncovering. One of the things there, of course, is single cell data is a snapshot in time.
Yes.
Right. And these are dynamic processes too. So it would be intriguing to see, does this change at different snapshots in time?
So the purpose of this piece of work exactly is that, can we extract dynamic information from snapshot data? So here is really, first of all, our input is not just the snapshot expression. We have estimation of instant velocity. So, but discrete, right? How can we connect them to get a full dynamic picture?
So the idea is because they're all in different stages, the cells, and in the process that you're getting the temporal aspect through the whole genome.
Right. So
okay.
Right. So you have all those snapshots. So then the question, you connect them to get, you have all the frames, right? Then you connect them to get this full the movies, right? You can do it.
Understand now. Thank you.
Yeah. So definitely later I'll show you the real lifestyle imaging. If I have, probably I don't have time for that, but I may point out that. So now I talk about this Jacobi analysis. Just keep in mind, we have this analytical function form. So we can do further analysis. What is shown here, this is high orders, differential geometry analysis. The one thing is this called acceleration field. So it's really the velocity, the derivative of velocity, right? And also the curvature. So the idea is that think about that you follow a cell, they have this trajectory, streamline, then you can, this 1D manifold, you can characterize as a velocity, acceleration, and also the curvature is how band it is, right? So for me, I really learned all this when I was a postdoc. So I read this kind of, it's in soft matter physics, and I have no idea I would apply this to cellular process. So let's give you some, why this is give you the new information. Well, let me just use the intuitive one. Suppose you drive a car, right? You drive your car, you can change the state of the car by several things. You can just press the pump, press the gas, and you press the brake or turn the steering wheel. So then the cell state will change. But we know when you do this, the cell state actual change, but takes time, right? It happen later. So if you can identify when this acceleration fields or those curvature things, really try to identify the moments you make those manipulation, and then what effects later time. You can really can identify those events in very early stage. So let's use this example. What is shown here is the two genes, right? They say this is starting from a naive cell differentiation. If you want to study this differentiation process, you may have, let's say you have two markers, marker gene, you have fluorescent labeling of them. Then you follow it, you find, oh, okay, there's one marker gene, the green, the red fluorescence signal turns on. Then here, oh, see those cells committed to this phenotype, right? That's what it's in. But actually, the cell make the commitment much earlier, before the signal turns on. So if you look at here is the acceleration field, you see the large magnitude part is here. Why is here? Well, you notice what happens here is, so here is, you look at this, the acceleration vector, so you have two components. So this component reflects that X2, the gene 2 self-activation, and this is gene 2 inhibition X1. So what happens here is here. In this, this model sim, I do use a Hill function. So you have a threshold. That's where your gene 2 reach a threshold that can have this self-activation and inhibition. So that's why you see this sudden acceleration. And then if you use the marker, really the marker, the gene expression, you read out much later time.
Come here.
Yeah.
Come, I'll take you soon.
Yes. Is there a question?
Yeah, I think that might have been a mistake.
Okay, yeah.
Thank you.
Okay, so this toy model shows you how this acceleration analysis can identify the events earlier before you have detectable markers. So here's one example, real example. This is zebrafish post-embryonic development. This is the single-cell RNA-seq data represented in this UMAP space. And this is the vector field. You see those are attractors corresponding to differentiate the states. And this is progenitor cells, okay? So this is corresponding acceleration field. Then you see those acceleration, high magnitude acceleration precedes those cell commitment, as you expected. You look at those genes have high acceleration. you do a geo analysis, you figured out those genes really specify those cell phenotypes. Especially, let's look at this group of cells. In the original studies, they could not tell what cell type to go. Probably just because they stopped experimenting early, so the marker genes have been turned on yet, so they cannot tell our gene. So we look at this. We look at the genes have high acceleration. Then we do a geo analysis and realize those cartilage chondrocyte, cartilage chondrocyte. So this analysis we can predict those group cells eventually will differentiate into chondrocyte. Another analysis is when we have this vector field, we can study the transition dynamics, especially when we do reprogramming or when we change phenotype. We want to know how can we efficiently convert one phenotype to another, or in some medical practice, you really want to really slow down or abolish that process, right? So what we do is, from one phenotype to another phenotype, there are many possible ways from one state to go another. Just like you have two cities, so you can drive from one city to another one. You can take a highway, you can take many local ways. Clearly, the highway is more efficient, right? So that's the most likely the cell will take. So what we want to do is, you give me two states. I want to find out what's the most likely path will take. So then we can try to manipulate. So mathematically, it's really for each pass you can assert with the probability, and so it's really you may realize essentially Feynman pass integral. Then you try to find the state, the path with a minimal action. So this least action, that's the most probable path. So mathematically, you define action like this. You try to find what's the minimize it, find action. So you find it's minimized along the trajectory space. So you can do this. Then here I show you for this process. So for different phenotypes, this is reprogramming those least action paths. For each path, you can look at the gene expression along the path of the genes. Especially look at those genes very highly correlate with the path progression. And then those transferrin factors most likely will participate in the gene regulation. So we identify them as potential reprogram factors. Indeed, for this process, right, for many of these cell types, they really have already studied experimentally. So we notice those identified top regulators.
Can I ask, what's the x-axis in that transcriptomics kinetic?
Yeah. So this is the least action time, really the path, right? The contour length of this path. You can do that.
Y-axis is the path.
Y-axis is the path. This is each one is really many genes. So each line is how one gene change along the path. So you have some genes, they will see this is high, then eventually persists for a while, then goes down.
And how is it clustered?
What's that?
How's it clustered? What's your x-axis?
I think this has been done, this hierarchy clustering to make it easier for visualization.
Okay, so but you've got in your Tisney-like thing that you're transitioning between the different states there, the new to the meg. Where's new and meg on that figure on the right?
Sorry, let me see what's your
you see what I mean, like in your transdifferentiation.
Okay, this is yeah. Oh, for this specific example I shown here is actually this is a differentiation process, the stem cell to BAS. That's what I show. You can definitely show others like a MAC to new as well. Right?
Sure, so that's HSC.
Yeah, this is the HSC to BAS. So
I don't see that path there. Where is
that?
Yeah.
I think I should replace this with because what is shown here is the reprogramming, really from differentiated to another one. Right. This exactly. What I show here is that spontaneous process. But you can do same thing for this for the reprogramming process, right? So
is that what it does? Is it straight like that?
No, it's not straight. It's not straight.
All right, somebody just.
This is high dimensional. First of all, it's high dimensional, right?
No,
And
sure. I guess one of the things I'm intrigued about is, right, you're collapsing high dimensional data down to get this transdifferentiation sort of map. And you're using the trajectories between the different states in that map as an axis, your y-axis, and then the genes that move you along that axis in the x coordinate, right?
Yeah.
And I'm intrigued what that looks like for the different connections and what insights that gives you. It looks like there's definitely some structure there.
Yeah.
You know, is that structure along that high dimensional axis that you're drawing, or is it just refactoring of that, a reclustering of that? Do you see what I mean?
So let me see if I can clarify this. So this LAP, LAP time, you can say, so because from two states, you have this one-dimensional manifold that connected, right? So this is like this is called the most probable path. So all this really plotted along this one. So LAP time really the contour length of along this path. You can view that, right? And then this is the genes, because this path is this in this high dimensional gene expression space. It's a 1D manifold in the high dimensional space. So this really shows all those components for each degree freedom corresponding each gene expression, how it will change along this path.
Agreed. But I guess my point was, is the order meaningful here in the x-axis or not?
The axis, you mean the x-axis? So each one is just corresponding one gene.
It's just one gene, but there's no order that it doesn't mean anything.
No.
Okay.
Well, I think we just plotted, we use hierarchy to make it, try to group genes a similar property together.
But yeah, that was my point. Was I wondered if it was then going to highlight the gene dynamics that were important in that process. And then one could build a profile from the cell state transitions to the gene dynamics. Do you see
that's exactly what we we try to utilize this. For example, here I show this highlight here, this carbon GATA1, right? GATA2. So that shows you how these genes clearly there's turn on at the later right? So that means at early stage this gene doesn't play much role, but later then it turns on, right? It participates in the regulation. There were gene early event. So by having this, the key thing is now you have those. Suppose you want to reprogram. Now I really want to accelerate the process. So with this, you will have idea what are the genes you may manipulate, and even at what stage. You may in another state to turn on all the time. Some gene need to be turned on early, some turn on late. Some gene need to be only turned on transiently, right? If keep constitutively turn on may not be necessarily well, right? Think about system supposed to make a turn. Then if you push, when the system goes from left to right, if you push it, then it's helpful. But then make a turn, it still continue to push. Then it'll be resist, right? It's gonna slow down the process. So having this information will help us to design how the strategies we can accelerate the reprogramming process, for example.
Okay, thank you.
Yeah. Okay, and let me see. I think how much time do we have? Do I have?
Well, you have technically another half an hour you want to keep going.
Okay, well, I think I'll just briefly, since I think I'll focus on this the single cell genomic data, but I briefly mentioned, well, in that work, what do? We have snapshot data. We try to recover the dynamic from snapshot data. Well, there, I have to say there's certain limitations, right? You have some assumption how valid they are. That's a question. So we have another route is we use lifestyle imaging to do it. So what I show here is mainly a series of papers done by my former postdoc, Wei Kang, and together with a few graduate students here, even undergraduates, Yaxuan. So here, the challenge is different because when you do lifestyle imaging is, okay, still you want to trace the same cell over time. In this case, you need to keep the cell happy. And the observable, the choice of observable is even tricky because you cannot really monitor many things, right? Because a lot of constraints. Say for labeling, you have this phototoxicity. So there, we work out a way is we look at the collective modes. That means the collective variable, like the morphology or cell features, are being already routinely used the pathology. Then we use this instead of just as a statistical variable, really dynamic variable, help to specify cell state. So briefly, I want to show you is what we do is we have cell, like this EMT. We have from this like epithelial cell to mesenchymal cells. They have dramatic change in this morphology and also gene expression. This is fluorescence for channel for Remington. We have endogenous labeling. So what we do is we need to quantify those images. We develop a machine learning technique to segment cells, which I won't go through in detail. Then the thing is, you got this image, you quantify them. So here we use this active shape model. Essentially, we use a vector to represent, math vector represent the cell morphology, cell body morphology. So you have this vector in this shape space. So you can even do principal mode analysis to get a basis function, similar like before we have a basis function. So then within this space, any shape can be represented a linear combination of this basis function. So you have a mathematical representation. So now you have this mathematical language, right? So I show you, we have this EMT process. We do live cell imaging, we monitor. So the reason why we do this, the morphology cell features is we have great expand the state space representation. Therefore, you see, at the beginning I talk about the ZDT, right? Here, Z in this case there's a feature space variable. So we have this live imaging. We can do extract live imaging track cell feature. We got this represented in the state space, well, trajectories. Now we do have a single cell trajectories over time. Then we can do analysis. So show you here example. You have a cell, this is one cell, 2 days continuous imaging. So this is cell shape, the yellow is Remington. Then from this image, we can extract, say this top is morphology, bottom, we got this morphology, we got this Remington, this texture features. It's high dimension. I represent in 2-dimension space. You got this single cell trajectories. Just like you have a bow, you have this movement, you got the trajectories. Now you got many, right? You got many trajectories. You have clear, you can show you have 2 types. You got 2 types of trajectories. And then you can do clustering to look at the similarity of the trajectories. You use this dynamic time warping, really try to match them. You see they have 2 types groups. What I'll do is you have the trajectories. Then like in chemistry, you have single molecule trajectory, right? So now you can do analysis. You ask the question, how my cell change from one phenotype, that's one tractor to another tractor. Then in mathematics, there are a lot of studies in the field of rate theories. And I think what I'll do is I'll just skip a lot of details because I want to show you can find out equation motion. You get this from the data, but I want to really highlight something. The key thing is from our data, we figured out there are 2 types of trajectories, and we from data we work out there's the reason, mechanics understanding of this, what happens. I use this system to schematic illustrate what may happen. So you have a tractor. So the system, what happens, you have add TGI beta. Then the cells, then this epithelial phenotype got destabilized. So then there's new phenotype emerge. In this state space, you can see you have these 2 attractors, then you have saddles. So when you increase TGI beta, when it's increase TGI beta, your epithelial attractors become less than stable, eventually become unstable. Then that's through this kind of, you see the saddle merge. Then you see this become downhill, right? Then this one further increase, then this one become downhill. You got 2 paths. Okay, so our lifestyle imaging, we got this we got these transitions. We show there are two types of trajectories. Why I got excited? Because my, I should probably just start from here. So this is because reason in this EMT study, we have an open question. The open question, as shown in this review paper, is we ask, one cell change from absolute to mesenchymal cell, it have to take some continuous path. It's just a one-dimensional path, or there are multiple paths? So this is the open question. Then in the field, a lot of pseudo-time analysis. The pseudo-time analysis means what they do is they have expression, single cell genomic data. They look at the similarity, then align them. So this is one study using this monocle analysis on single cell R6 data. So they show this is just boring, from absolute mesenchymal just continues to one-dimensional path. And this is the proteome data. Also, they use the pseudo-time analysis to say, okay, from absolute to mesenchymal, there's one path. Well, go back, you have another path. This is characterized for a characteristic for this system, as an open system. So, but our lifestyle imaging, we found two paths. Interestingly, my students also analyzing single cell R6 data using this vector field, because now we have vector field. So this pseudo-time, you really look at the mean trajectory, right? But this is a single cell sequence vector field analysis. We can run trajectories. We look at those. My student Sophia, she found also two paths, similar like what we got from lifestyle imaging. And she further, we want to understand what happens. So we analyze in the system, we do kind of a coordinate rotation. So what is shown here in this is the x-axis, the cell cycle progression. Think about that, from left to right is cell from this G1S, G2M, then divide, then repeat, right? You then go back, repeat. So this is the another degree freedom that's reflecting cell cycle, the EMT, right? So what you see here is this is the cell progression, cell cycle progression. Then you see at here, this is G1, G1S. There's a cell move along this EMT axis. This is the G2M. So this is G1S arrest, G1S arrest, G2M arrest. But what surprised me is this. In her study, in her data analysis, she showed this. My first response is something must be wrong. It doesn't make sense, right? You think about cell cycle, always a progression forward. So this one suggests that, some go backward, right? This go backward, and here you have cell go backwards, go to G1S. I think, I told her, well, something wrong. We have to figure it out. Then I talked to Wayne Stellers. So he has a very nice work. What they do is in recent work in this bioarchive preprint. So they have done a lot of multiplex staining study and also lifestyle imaging. And what they report, actually, they found this. The cells exit the cell cycle from G2 at G2M arrest. And then they found those cells really converge to a pseudo G1S state. So they did observe this, and this is consistent with what we Sophia just observed. So this is what really excited me. So I just mentioned, see, this is a traditional, this is pseudo-time analysis. They do only get average information, so miss a lot of details. Well, now we show this both lifestyle imaging and is our new vector field-based analysis. We can get more detailed information. Okay, so what I want to summarize here is I want to show, in the past few years, my lab is focusing on is we want to get genome-wide and gene-specific quantitative, semi-quantitative, the dynamics information. And we use, it's all based on data, based on the single cell snapshot genomic data and lifestyle imaging data. So I think that's what's exciting is, traditionally, with a lot of data, data science, right, to do statistical analysis, we try to place all this analysis within the framework of dynamical system theory. So what we want to get is, you have a cell. We know you have input, stimuli, you got output. We ask a question, it's a very ambitious question. Can we really work out a set of equations. Now, if you just tell me what's the stimuli, I will perform in silico simulation. I will tell you cellular response. Like you have a drug, you have combination drugs, I can tell you what the drug response. That will greatly simplify our study and save a lot of efforts in drug discovery and many other things, right? So this may sound like very basic, but I do want to remind you what happened in parallel at molecular levels. That's the molecular dynamic simulations. When those guys, they start this molecular dynamic simulation on peptides, proteins, half century ago. Those are very primitive, but after many years study, now molecular dynamics simulation have been very important part as a quantitative tools for us to do drug discovery and many other things. So I really vision and believe that with continued development, we can also place all the cellular dynamics in the context, in the framework of dynamic system theory. So with this, I want to just conclude by acknowledging the people who really do the work. I already highlight the researchers, some research like Yan and Xiaojie. They are the real drivers for the genomic, the first project, and Wei Kang and other Dante, Yaxuan, and Thomas on this. The second work. And also, I said, this is really collaboration among many researchers and also the funding agency. With this, I'll just stop here. Okay. Thank you.
Yes, thank you very much. This has been an excellent presentation. Thank you. And beautiful visuals, really nice data-driven research, nicely grounded in systems biology and dynamical systems. At this stage, I know that Sandy has asked some questions. I don't know if other people in this room, virtual, might have other questions. I know we have systems biologists and bioinformatically minded people in the department.
Okay. Yeah, I see Daniel Glacier asked question, how were the genes clustered?
That's right.
Oh, that was with the discussion with Sandy.
Yeah, okay.
Right.
Yeah. So I just mentioned, I think we have done clustering.
Yeah.
So in terms of whether research is, this is a really nice way to obviously present it. What's not clear to me is what are the next steps? Where are you taking this to? How are you actually impacting the work that your biological collaborators are doing?
Yes, that's right. So as a dynamic system theory researcher, I said we are interested in the dynamic equation motion, right? So I've shown you how we can do it from snapshot, or we got the lifestyle imaging. Now a major push for us is to integrate the two, because both have the good thing is the first one, you got the gene specific information, right? This is where biology care about. How each gene regulates each other. If you haven't talked about regulation, we can do in silico perturbation and predict how this mutation perturbation will change the dynamics. Mm-hmm. But then the limitation there is, the dynamics inferred, right? Is that right? Right? We have to test against the experimental thing. But then there's lifestyle imaging. The lifestyle imaging, the limitation is, I said there's, the number degree freedom and things. So we decide the goal is go to this collective feature space, which, right, this is the right, I think this is the right way to go. And also, especially if we think about dynamic system, really typically lie in low dimension manifold. So we can use this composite manifold. We can, we haven't proved it. We generally believe that we can faithfully represent the cellular, how the system evolve along this dynamic manifold, right? We can reconstruct dynamic manifold. But the question is, that's in composite cell feature space, we need to connect to the gene expression space. So there, so that's system biology or biology, they care about is the gene regulation. So by combining the two, we'll get the full picture, the dynamics in the gene expression space. We now we have the model, similar like we have done in system biology, we have the network model. We can predict, we can test.
Yeah, that sounds very ambitious, but I think that you guys are in definitely a good position to go for that. So I'm just going to start assuming that people who are sharing their screens are interested in asking questions. And I know that Noemi was first. So Noemi.
Yeah, thanks. That's definitely a worthwhile aim regarding your last comment to integrate live cell imaging with single cell sequencing. They're very powerful techniques and complementary to each other. And I know there are some efforts also in the biotechnology domain to integrate the two, but then the computational domain can bring something to the table as well, I believe. I did want to ask about, at some point you mentioned pseudotime inference methods that predict cell cycle progression from single cell sequencing. And then I was intrigued by the result you showed that how you essentially could quantify the skipping of mitosis, which listed is a well-described phenomenon to some extent.
Yeah.
Yeah, exactly. So I was wondering if you had any thoughts how these pseudotime inference methods have to be changed, or maybe just the parameters have to be changed in order to capture this process, and to what proportion of cells it applies.
So as I learned from Win Stellar, they have done a lot of so those cells, they found those cells G2M, so already have DNA duplicate, right? Then they skip cell cycle, then somehow they will kind of degrade some of their cycling and go back to this pseudo G1 space. So that's one mechanism to form this polyploidy, right? This is then with this definitely we're still working on this, right? And there we can have these trajectories. We will be able, just I mentioned for the reprogram, there we'll identify those factors that's participate in the regulation of the process. Then we can manipulate, right? So we can control the system go either here or escape state, or even go other states, right? The goal, ultimate goal is we want to control the cell.
Yeah, but this is quantified from live cell imaging, though, right?
Oh no! Oh, this is from single cell RNA-seq data. Oh
okay.
Yeah,
okay.
Yeah. This is from single cell RNA-seq data.
So if I want to find out more details about how this method is different from the pseudotime inference, I just need to read that preprint, right?
Yeah. So which I didn't show up in actually their slide I didn't show. So how we do this? Actually, this is you notice this one. First of all, my student just do it. This is do the analysis in this UMAP space. You have data. Typically, do a UMAP dimension reduction. Then she analyzing this. Although trajectory is simulating high dimension. So when I saw this, we see one show here is the top two, because I know the system, their cell cycle. I want to really figure out how the cell cycle involved in this. So then I realized one thing, there's a problem for when you do dimension reduction with generic dimensionism, because we know epithelial cells, there should be, it's not just a attractor, fixed point attractor, really a cell cycle, right? You should see a cycle. But what happens, because this variance, right? All those dimension reduction based on variance, those variance due to cell cycle of epithelial cells is kind of overshadowed by the difference between this epithelial mesenchymal cells. So the detail now shown here. Therefore, what we did is we simply for this one, I simply use there's a paper they use called Revleo. What those guy does is you have a PCA space, right? You just do a rotation. You try to rotation to get a new subspace. So the new subspace, the leading to the component of the subspace has maximum projection of cell cycle. So you kind of that's what we got this leading. So this is the cell cycle coordinates from the leading to cell cycle PC space. No, not PC, right? Now it's a dual rotation. So you got that degree. Then we got this other thing. That's how we can really see how this EMT and the cell cycle are coupled.
Okay. So you're separating the spaces first.
Yeah. So I think the lesson we learned is that when we do biology data, we're talking about biological system, right? So we know thinking about the biological problem is very important. If you just blindly just apply those algorithms, we may miss some very relevant to biology.
Very true. Very cool. Thank you.
Cool. Gosia, I think you have a question as well.
Yes, I do. First of all, very, very beautiful, beautiful talk. As a person who comes from dynamical systems background, I just can't help to, to be very excited about seeing all the vector spaces and phase plan analysis. It's great. My question is about more of the machine learning / data component of your work. And I'm wondering how your work is going to be sensitive with regard to noise in data and potential problems with reproducibility, because it's the way a lot of us are, putting data on the shared resources. It is a little bit questionable. Sometimes some cells may be missing some information. Sometimes data will not be having all the necessary gene expression. And I'm wondering how tolerant to these kind of errors is your work.
That's a great point, right? All those things really heavily rely on the qualitative data, right? If you do not have quality data, you do not expect to get a meaningful results. What all your concern kind of all justified, valid, and we have to keep that in mind. That's thing I can say. So then, even beyond that, right? I also mentioned, we have to realize when we do this, we have a lot of assumptions. Like the first one I talked about, the main assumption is this Markovian dynamics. That's really questionable. Think about it, right? We only have transcriptome, we have proteins, we have epigenetics. They all have probably even slower dynamics. How that will affect your results? So all those are still, we need to explore. But my general current theoretical picture is, as I mentioned, a dynamic system is typically in a very low dimensional manifold. So I think that's the key thing for us. We can either use this genomic data, just transcription data, or even our lifestyle imaging. We have some feature things. We assume we can kind of more or less completely to represent cellular dynamics. I think this is a very key part, and may not transparent to if a biologist or something, right? To think about how is the genome, right? We're talking about genome of things, and you have epigenome, you have proteome, and then you only have a partial information. Is that sufficient? I want to point out one thing. I got very, very impressed as I read the article. They talk about the machine learning. You know what? So they show people now can learn how a people looks just by the voice. So why this struck me is really show us, and that's exactly I mentioned that our real in nature a dynamic system always over like a very low dimension manifold. So even that means if you only look at it from some subspace, some angle, somehow that can already reflect the system dynamics. So it's the projection. Which other word you can say is dynamic manifold means if you project on subspace, the dimensionality probably similar. So you have not much degeneracy.
Gotcha. Thank you so much.
Cool. Sandy.
Yeah, thanks. That was definitely lovely talk and really well explained. A lot of complex stuff there, and I appreciate you taking the time for some people like me that are a bit slower to understand, to elaborate a little bit more. I really liked your sort of grand vision statement at the end with, can we sort of synthetically, predict drug responses and so on without having to do those experiments in the future. I guess that, one of the things that we've spent decades trying to do it in IMO is understand the importance of interactions. And so cells communicate with each other, that communication changes their behavior and potentially then changes the context. And so an obvious question seems, is that something that this framework could also incorporate, which is cell-to-cell communication?
That's a great point. That's a great question. Yes, definitely. Here I only talk about single cell. Well, when I write down the equations, I should have really, this Z is talk about one cell, or really a group of cells that have to have this A, including the cell-cell interactions, right? Or can use the cell-cell interact as an external variable. So the framework definitely expands, right? So this is a starting point only to look at a single cell. So because of my background, so in physics or chemistry, talk about particle, many body problem. You do have a strongly correlated, there's particle interaction, all things. So you can view what I'm showing here. I have done some kind of mean field approximation. So got the single particle equation motion. I can expand it. I can, especially if I have like say spatial genomics information. I have all those. So I can add on this cell-cell interaction terms. Then yeah, what exciting exactly is because my background, right? I have used to those kind of language when I talk about the particles, molecules. I feel I can, apply those kind of thinking in cells, right? So this is one thing I didn't expect. When my background in chemistry, theoretical chemistry, then I go into biology. I thought what I have learned, have been probably won't be used. I have learned in Hilbert space from quantum mechanics, other things, past integral things. Then surprised me, many years later, I started to use the same kind of tools language to study cells. And similar like, to many particle physics, right? Things we can apply this. I think this is what got me excited.
Great. Yeah, I think my point there was, there's new, and I guess, single cells exploding, but so is also spatial transcriptomics.
Yeah.
And so is there a way to sort of integrate single cell understanding with spatial transcriptomics and somehow calculate interactions as a result of, we know how they behave in isolation.
Yeah.
We know how they're behaving in context together. Can we infer interactions and dynamics somehow? Yeah.
That's exactly, I mentioned is you can have write down this, a single cell, then you have another cell, right? You can like agent-based modeling. You can think about that, right?
Exactly. That's one of the things we think agent-based approaches could be useful here.
Yeah.
So, there might be a cool opportunity to embed some of these networks in our agents, right? That's something we should talk about. Anyway, thanks for your talk.
Yes, thank you very much. I assume that we have had enough questions, but if anyone has any burning one, please, this is the right time. And if not, once again, thank you very much for your lovely presentation. It was great. We learned a lot, and I think that you might have inspired quite a few of us to actually look beyond our agent-based models into this more dynamic systems approach to modeling cancer. And this is
I'm well aware about you guys' work.
Very good. Well, thank you very, very much.
Oh, thank you. Thank you, everyone.
Bye, everyone.
Bye.